All right, so let's go ahead and pick up where we left off with the last lecture. We're talking about the key phylum or groups of fungi that we're going to be looking at. And as I mentioned in the last part, the mycota means it's a fungal phylum. So zygomycota, tritiomycota, etc. Hopefully that makes life easier when we're trying to remember these things. Mycota is a key term. So let me just underline it just to reemphasize mycota, mycota, mycota. Okay, make sure you guys remember that. So, all right, now let's start looking at fungal features. When we look at an organism, how do we know it's a fungus and not an animal, a plant, a protist, a bacteria, etc.? Well, we're looking for some kind of these distinguishing features. And one of the distinguishing features that helps identify fungus are these things called hyphae. I'm going to put this in quotes. Hyphae are these tiny little filaments that extend out from the fungal body, and they absorb the nutrients. They secrete the enzymes, absorb the nutrients. But that's really a big important fungal feature, is that we look for fungus, we look for hyphae. So how do you know if you have a fungal infection? Well, can you see the fungal hyphae? Are those structures present? Things like that. But that is somewhat of a defining characteristic, not specifically unique to fungus, but an important feature of fungus. Now, the hyphae come in a couple different types or categories. So this is where things get kind of goofy here. A lot of hyphae will have these little things called septa. So the septa is a dividing wall in the hyphae that forms cells. So you've heard of a thing called a septum in your nose that divides the right and the left side from each other. Well, lots of fungal hyphae or fungus will have hyphae with little septa or septum. There it is right there. And it's just this little dividing structure. Now, often the septum have little holes in them or pores that allow nutrients and things to go through from one hyphae cell to the next, to the next, to the next, and so on. So if there are nutrients over here, let's say that's a piece of bark on a tree, a dead tree that the hyphae is secreting digestive enzymes on, and as those nutrients break down, they get absorbed in the hyphae here. They then can go through the little hole or pore in the septa and get transported throughout the fungal body. So that way the rest of the fungus can share in the nutrients that certain hyphae have found. So now here's where it gets kind of goofy. Some fungus are what we call a coincidic fungi. So a coincidic fungi this is goofy and weird. They lack septa, so they're basically one big giant cell with a whole bunch of nuclei inside of them. So this is where it gets hard to talk about fungi genetically. You know, if you have all of your genetic material, you're what we call diploid or 2N. If you have half your genetic material, you're haploid or N. Actually, let me move this down here. 2N is diploid. Sorry guys, I can't draw this out real well. Diploid. Half the genetic material, haploid. And we represent genetically a diploid by 2N, a haploid with an N. Well, coincidic fungi, what do you do here? there's a whole bunch of nuclei within this one big giant cell. So this again is where it gets weird, and we go, oh, okay. Well, we call it N plus N. All right, it's a term called dikaryotic. We're going to talk about that in a little bit. I wanted to introduce it here with the coincidic fungi, and then we'll continue talking about that in a little bit. So, okay, so a couple key structures or features that help us identify fungi. Other key things, the mycelium. So when you get a big mass of connected hyphae that comes together, then it forms this thing known as a mycelium. So I want you guys to go out, and if there are some leaves that have been sitting on the ground for a while, since last spring or for an extended period of time, a couple months, lift them up, look underneath. Do you see this mycelium? But be careful. It might actually be a slime mold. So how would we identify the difference? Structurally, superficially, we look at them and they may look very similar, but we could determine for sure if it's a fungus by examining the cell walls. And remember, what is the key polysaccharide found in the cell walls of fungi? Cell walls are composed of blank. I want you to fill it in. I'm not going to tell you the answer. I mentioned it earlier in the previous part. If you're drawing a blank, just back up in your notes, look it up, fill it in here. But that's another big key feature when we're discussing fungi, is the mycelium and the hyphae, their cell walls, are composed of a specific polysaccharide. All right, other things to mention here. When we get that hyphae or you get that fungus growing and it's doing its thing and it's saying, okay, time for reproduction, then it pops up and produces these little things we call the fruitine bodies, the spore-producing structures. So the mycelium tends to be, if we're talking about a fungus in the soil, tends to be in the soil underground, and then it will shoot up some of the hyphae to produce spores. And we'll talk about the difference between each of the phylum really is in the spore-producing structure here. It's one way we identify phyla. A fungus is by looking at the spore-producing structure. So that's going to be used during asexual reproduction. There are no male and females here, but sexual reproduction or exchange of genetics occurs when the hyphae fuse and they literally swap pieces of DNA. And again, we'll get into that in a bit. But what we tend to see and eat, you guys had mushrooms on pizza, mushroom soup, mushroom in a salad, wherever you would use a mushroom, you're eating the reproductive structure, the asexual. Let me put that. The it's supposed to be an A. Sexual reproductive structure of the mushroom, or of the fungus, is what we tend to eat and consume. We often don't see the mycelium. So the good news here is if you have mushrooms and you're like, ooh, there's some morels or shiitakes or whatever mushrooms you eat, you can simply cut them off right there and leave the mycelium alone, and it can regrow. It can produce new mushrooms. So not a hard thing to do. You can actually grow your own mushrooms. There's a multi-million-dollar mushroom farm starting up in the Chicago area. Millions of dollars made growing mushrooms. And the awesome thing here, economically, you can grow mushrooms year-round. Corn, you get one crop a year. Beans, one crop. Mushrooms takes about 6 weeks. So every 6 weeks, you can have a new harvest of mushrooms coming in. So a lot of money to be made here. And what we're doing is we're trying to get the mycelium to produce a lot of these reproductive structures. And that's what we tend to consume. So, okay, other things to mention and some terminology here that I want to make sure you guys are comfortable with. When we talk about reproduction, spores are the most common means of reproduction. Spores are N or haploid. Oh, that's supposed to be a little n. Do not put a big N. Darn autocorrect. Haploid. It contains half the genetic material. 2N is what we call diploid. Oh. Full genetic material. And then again, we get that weird thing. No, lowercase n plus n is dikaryotic. And these are mushrooms that have 2 nuclei, but the nuclei don't fuse. So normally, the nuclei, when you're reproducing, you have half the genetics from each individual, and they fuse and they create one diploid nucleus. But some mushrooms are just weird, and they don't fuse their nuclei. So again, it's a weird world when we get into the mushrooms or into the fungus, and they do all sorts of goofy things. So, okay, other features to mention briefly here. Some of these are repeats. So they secrete digestive enzymes. The 2 key digestive enzymes are lignase and cellulase. Lignin is a fiber in wood. Cellulose is a fiber in plant cell walls. Fungi have the ability to break it down and decompose it. So this fungus right here is called a shelf fungus or bract fungus. Grows on the side of a tree. They are secreting digestive enzymes that slowly eat away the bark of the tree. So it can actually, over time, kill the tree and become problematic if it's too big. It can weaken the tree. And then they absorb nutrients externally. So we're different. We swallow the food, goes internal, we digest it, absorb the nutrients. As I mentioned before, fungus excrete enzymes and absorb the nutrients externally into the hyphae, and then it moves from one cell to the next, to the next, to the next for the fungal body to gain the nutrition. Some fungus are carnivorous up here. These little guys here, they are actually creating little loops where they're like little snares or lassos. And when these little soil invertebrates, these are little worms, go moving through the soil, if they swim through that little loop, it's like a snare, and it just tightens around the worm and traps the worm. They secrete digestive enzymes, kill the worm, and eat it. So carnivores. Some of them are little microscopic carnivores. And the other thing to mention, some fungal hyphae will actually have extensions that go into cells of plants, and they absorb the nutrients from the plant. These are known as haustorium. And we'll talk about these more when we get to the end and we look at relationships between fungus and plants and how they are, in some cases, many cases, symbiotic, and they're mutual. Some cases they're parasitic. That's in the bad category. So, okay, so some big general picture of what fungus can do and how they're organized and all these different traits and features. Now it's time to start talking about fungus. So I mentioned before there are 5 key groups. Yes, I'm going to talk about briefly a couple other groups and I will highlight the key ones I want you guys to make sure you know. So the first one, just to briefly mention, is the Microsporidia. I know we just briefly talked about it. This is not a group I'm going to hold you guys accountable for. It's just more of a introduction with a little bit of information. So then in the future, and I mean another course down the road, if you hear about this, you're like, oh, okay, there's a little bit of familiarity with it. Microsporidia are those intracellular parasites. They actually can get inside the cell and parasitize internally and create problems. Your immune system can't find them. So that becomes a problem for anybody who's got a suppressed immune system or compromised immune system. This particular fungal infection can be very, very deadly. So somebody suffering from AIDS, you get a fungal infection or this Microsporidia infection and can be, again, seriously a serious problem for a person. So, okay. So again, don't worry about focusing on this one. I just wanted to briefly mention it again so you guys say, okay, I remember hearing about it if it comes up in the future for you. So the first one to memorize or remember and make sure you have a better understanding will be the Tridiomycota. We'll get into that group as we go into our next part of the lecture.